So this is part 2 of the lesson on recombinant DNA. So last class we started talking about recombinant DNA and using that technology in the context of genetic engineering, designing organisms to fit human needs. If you remember, we were talking about the different steps to creating an organism using recombinant DNA technology. And we had 3 steps to that process. Step 1 was to extract the DNA from the cell. Step 2 was to cut and paste that DNA together. We used restriction enzymes with sticky ends to cut DNA out. The last step is once we cut and paste that DNA together, we have to put that DNA back into the host organism. So we've already taken out a piece of DNA. We've already edited it so it has the traits that we want. So now our job is to go ahead and get that DNA back inside the organism that it belongs in, so that organism now has the traits we've added to the DNA. So let's talk about a few methods to do this, a few methods on how we insert DNA back into the host organism. And I'm just going to go over like maybe 4 examples or 4 methods to do this. There are other ways to do this, but this would be a very long lesson if I talked about all the different methods, so I'm just going to talk about some of the most common. So I'm going to go over 5 methods of getting DNA in from one organism into another. 5 methods: Plasmids, Agrobacterium, Microinjection, Electroporation, and Ballistics, Biolistics. And each of these methods can be applied to a different group of organisms, and you'll kind of see that as I go through them. So not all methods can be used for all types of organisms. So the first one we're going to talk about is plasmids, and plasmids are probably the most important. It's probably the most used method of getting DNA into a host organism. Specifically, plasmids allow you to get different genes inside bacteria. So we're going to talk about an example of why we might want to do that. Well, at first let's talk about plasmids in general. So plasmids are circular pieces of DNA found inside bacteria. So when we look at bacterial DNA, if you remember, our DNA is this double helix shape. Bacteria have that same DNA, but oftentimes they have a piece of DNA that comes in the form of a circle. We call that a plasmid. And plasmids are important because plasmids can be taken out of bacteria and put back into bacteria. So that makes it really easy for us to take this plasmid out of a bacteria, edit it, and then put it back into a bacteria cell. So the method looks something like this. We might have a plasmid, this circular piece of DNA from a bacteria, and then we have donor DNA, so usually something like human DNA. And there's always going to be this gene that we want to put inside the bacteria. So what we do is we take the plasmid out of the bacteria and we take the DNA out of the donor organism. We extract those 2 strands of DNA, and then we go ahead and use our restriction enzymes to cut up the 2 pieces of DNA. Now this is very important. When we find this restriction enzyme, it must have these specific traits. The first is that it makes 1 cut on the plasmid, only 1 cut on the bacteria DNA. And it makes a cut above and below the gene we want to move into the bacteria. So if we look at our diagram here, our plasmid, our circular piece of DNA that we want to insert the gene into, we need a restriction enzyme that cuts it in one place that will allow us to cut it open. That same restriction enzyme needs to cut above and below the gene that we're trying to insert into the plasmid. So maybe on our human DNA or other organisms' DNA, we cut above and below the gene we want. And if you could see that, we would imagine it looks something like this. With the plasmid cut open, we have 1 cut on the plasmid that opens it up. We can insert the cutout gene we want right into that plasmid. And as long as they're cut using the same sticky ends, those genes will stick together, and that DNA will just fit right in there and become part of the plasmid. And then because of the way bacteria work, they naturally share these plasmids. Bacteria actually naturally take up the plasmid. So if we put bacteria in a petri dish, and we put that plasmid in a petri dish, the bacteria would just naturally take up that plasmid, and that bacteria now has the new gene that we inserted into that plasmid. So the bacteria will now start doing whatever that DNA tells it to do. So a perfect example of using plasmids or using bacteria is in insulin for diabetics. So people who are diabetic, they don't make the protein insulin that helps them regulate or control their blood sugar. So the question is, well, we know that people who have insulin, they take shots of insulin, but where does that insulin come from? They actually used to extract it from horses and use horse insulin, but now they actually use real human insulin as a medicine. But where does that insulin come from? We don't take it from other humans. So what happened is scientists took the gene for insulin and they cut it out of human DNA. They cut it out of a human that was making insulin, and they inserted that DNA into a plasmid, put that plasmid inside a bacteria, and now the bacteria make the human insulin. So these bacteria can be grown in a petri dish or something similar, and then they can extract the insulin that the bacteria are making and use that for people with diabetes. So this is a perfect example of how this technology can be used. This is a great slide that you might just want to pause and look at. It's got a lot on it, but it explains the process really well of how to get DNA into a bacteria. You have your human cell and your bacteria cell. You extract the DNA from the human, the gene that you want, and cut it out using a restriction enzyme. At the same time, you extract a plasmid from a bacteria, and you cut it open using the same restriction enzyme. And then the two sticky ends will join together, and that will be inserted back into the bacteria. The bacteria will read the human DNA and make the protein that got inserted in, or follow the instructions that got inserted in, follow that gene that got inserted in. So this is a great slide that explains the entire process of using plasmids. The couple other methods of using are kind of vectors to getting genes inside cells. This is a great one used in certain plants. It's called Agrobacterium. And Agrobacterium is a naturally occurring bacteria that causes tumors in plants. So you can see here this tree that I'm standing next to has this huge tumor on it, or these tumors on the roots here. These are caused by a bacteria that infect the plant. And the way that they actually infect the plant is they do it by injecting their DNA into the plant cells. And that's actually what causes the tumor. And many viruses work this way as well. So what happens is scientists can remove the gene that causes the tumor, and they can insert the gene that they want into the plant. So the Agrobacterium, instead of infecting it and causing a tumor, will infect it but insert the gene that we want into it. Just kind of a way of tricking that bacteria to infecting, but actually inserting genes into it that we want. A third method is called microinjection, and microinjection is exactly what it sounds like. If you're trying to think of a way to get DNA into a cell, well, why not just inject it? And that's what they actually do. They use an incredibly tiny needle. It's usually like blown glass. Has an extremely small, tiny point. It's so small that it can actually pierce through the cell membrane, and they can inject a DNA sample right into the nucleus of the cell. This is extremely difficult to do, and it's not commonly done, mostly done with animal cells that are harder to get DNA into. So this might be an egg cell that they're injecting DNA into. Electroporation is actually a pretty clever way. So this is again done with plant cells. They actually remove the cell wall of the plant. They call it a protoplast when you do that. And they shock this protoplast with electricity. What happens when you shock it with electricity, the cell membrane actually opens up tiny holes. And those tiny holes are large enough for DNA to enter. So what they do is they take the cell and they put it in a solution of edited DNA that they want inside the plant. They shock the cell with electricity, and then hopefully some of that DNA sneaks its way in. And then the last method of getting DNA inside a cell that I'm going to talk about, and probably the most interesting way, is something called Ballistics, which is when scientists use these tiny balls or bullets of tungsten or gold, and they coat those balls with the DNA that they're trying to insert into a cell. And then with these DNA-covered bullets, They shoot these bullets out of an air cannon, or actually in the past they used to use a shotgun to do this. They would shoot these bullets, very, very small, at the plant cell, and some of those bullets are small enough to enter the cell and bring the DNA with them. So they literally shoot DNA at the plant cells in hopes that some of them pierces the plant and makes its way in. So this is actually a pretty cool method. Again, this only works on plants. You can't do this with kind of human cells, but it works on plant cells. And you actually see here, this is pretty cool. This is you can buy the gene bullet online. These are the little tiny gold particles that are used to cover in DNA, so you can shoot it inside your cell. And just a milliliter costs $70 of these gold bullets, so pretty cool. So those are all just different methods of getting DNA inside of a cell. Again, like I said, there's many different methods, but those are some of just the most common.